Welcome to the fifth lecture in the course Mobile Source Air Toxics Risk Assessment. In the fifth lecture, we will focus on fate and transport of the air toxics, which will spread from air to water bodies to plants to people and to ecological receptors. In this fifth lecture, the main objective is to break down all the components on the fate and transport of mobile source air toxics. First segment, we have 5 of them in the fifth lecture, is to introduce you to risk assessment concepts. We will then have a second segment that I will take separate for media, the third for intake and ingestion rates, the fourth for toxicity, the fifth for human health risk calculations. Risk assessment concepts. This is just a brief introduction. We will see the process, how we conduct the fate and transport calculations. We will have to understand what is a scenario when we're doing risk assessment, how we will compute media concentration. And once we have media concentration calculations, we will talk about ingestion. Ingestion of the media for which we computed the concentrations. For example, plants will have tissue contamination. We will compute what is the ingestion of those tissues, and at the end, we must assess a dose. Ecological receptors, I'll make just a brief mention on ecological risk assessment. And remember, we have an entire lecture 6 for human health risk assessment, lecture 7 for ecological risk assessment. The process for the risk assessment starts with the dispersion modeling. We then have to pass information from the air dispersion model to tools that will import the results from air dispersion models into the risk assessment model, and then we need to import maps. Remember that both dispersion modeling and risk assessment, they are spatially distributed. For example, wind will blow in all the 360-degree directions. Pollutants will be transported spatially, therefore need to know what it is hitting, where the receptors are. For that, we need to import maps. We need to know where is the water body, the watershed, etc., using maps. Once we have the maps, we locate watersheds and water bodies, and we identify where sensitive receptors are located. Now, once we did the first phase on setting up the risk assessment, we must specify the emission rates for each one of the COPCs. And I described this in the first lecture, that COPC stands for Chemical of Potential Concern. We then define scenarios. These are the input scenarios, and we specify site parameters. Scenarios, we will see, these are the type of receptors that will receive the impact. For example, a farmer will eat its own food that they grows, and then if the soil is contaminated, the plants are contaminated, the milk that he may drink is contaminated, the eggs are contaminated, all will work as load to the farmer scenario. We will estimate process risk estimation, and that will define then the risk. We cycle to validate. Example in point. Sometimes emission rates are off, and we produce risks that make no sense. They're maybe too high or too little. And when we finally get results, we must analyze the result and rerun elements in the process that we will identify require revision. Once that's all done, we print the reports. This is part of the process of executing the risk assessment, where we need to know from the air dispersion model how the toxics will move from air to water, to soils, to plants, animals, and finally hit human beings. Same thing for ecological receptors. Now, the human health risk assessment calculations, in theory, the concept is simple. We can have a source, which could be point sources from stacks, or could be road segments, vehicles on those roads. Then many pathways, air, water, soils, plants, and it hits a receptor. And we need to compute cancer risks and non-cancer risks. This fifth lecture is focusing on the various pathways. How does it go from one media to the other one? These exposure pathways that we need to compute using fate and transport equations is composition and concentration on soils, concentration in water of all the chemicals produced. These will be things that are related to tomatoes, lettuce, beef, milk, pork, chicken, eggs, fish, and direct inhalation. Since those are things that will impact either the human being or the ecological receptor, we must then use fate and transport models to transport these chemicals through all these media. So it's a multimedia pathway to hit all these 10 types of exposure. Now the scenarios we would have the resident, and you can see here this resident has a carrot. But we consider that even resident will grow maybe tomatoes, maybe carrots, some produce, some lettuce. I know a lot of people that do. The second scenario we have here, it's the farmer. We consider that it's a self-sustaining farmer. You can create other scenarios where it only produces and drink its own milk, but no pork. We can change that. But the complete farmer, for our analysis, will ingest own-grown milk, eat pig, pork, eat chicken and eggs, their own produce, and fish. The fisher will only fish on the pond and eat the fish from the ponds or small lakes and get contaminated by the fish. Everybody can drink water from that lake, filter water, and drink the water. All these three scenarios will have an adult component and a child component. We need to compute them separate. Scenario for the adult resident, we already saw that the exposure will be direct inhalation, drinking water, eventual soil ingestion, and above ground produce, growing its own fresh veggies. Scenarios for farmers, it will be expanded by production of beef on top of the resident, the beef, the milk, pork, the fish, the eggs, and the poultry. And then the fisher scenario, it will only have the same as the resident plus the fish. If you have questions, do not hesitate to email me.